Hi everyone, my name is Nurika Hariastuti from the Center for Research and Development for Biomedical and Basic Health Technology under the Ministry of Health, Republic of Indonesia. In this video, I will share a study that we conducted about the applications of Freeze-thaw harvest for SARS-CoV-2 PCR external quality assurance panel material. Severe acute respiratory syndrome coronavirus 2 was first discovered in 2019 in Wuhan, and soon after that, this virus spread and caused a COVID-19 pandemic. In Indonesia, SARS-CoV-2 was first detected in March 2020, and at that time, the laboratory designated as the testing laboratory was only the virology laboratory in the Center for Research and Development for Biomedical and Basic Health Technology. As we already know that Indonesia has a high population and is widely distributed with various transportation complexity. To prevent delay in diagnostic, the government has appointed about 830 laboratories to carry out SARS-CoV-2 detection using the real-time RT-PCR method. While there are many reagents on the market and have not been fully validated, quality control is very important to ensure that the results issued are accurate and reliable. The quality assurance carried out by the laboratory should be in the form of internal quality assurance and external quality assurance. To evaluate the performance of testing laboratory, the Ministry of Health conducted the External Quality Assurance Program. In Indonesia, the External Quality Assurance Program for COVID-19 testing consists of confirmations or validation tests. In this program, the testing laboratory sends their tested samples to National Reference Laboratory to be confirmed. After that, the testing laboratory in the network can join the proficiency test, which they will accept a panel of specimens to be tested. A proficiency test panel commonly prepared from parallel culture, related to the need to provide a homogeneous large number of samples. SARS-CoV-2 has been successfully cultured and propagated using cell culture, both primary human airway epithelial cells and feral African green monkey kidney epithelial E6 cells. To extract cell-bound viruses, a combination of freeze-thaw or sonication is usually used. However, due to the purifying process, the virus isolate usually do not contain ribonuclease protein or RNP as in clinical sample. As a result, many testing laboratories submit invalid report in the proficiency test organized by the Ministry of Health and IHRD. This invalid status referred here is an invalid sampling process, not the result of the examination. Many laboratory participants encountered difficulty in determining the test result. This is because the RNP CD did not appear from the panel test and caused many laboratories did not to obtain a perfect result in the test. RNP in COVID-19 testing is an internal control for specimen collection because it comes from the epithelial cells. In viral cultures, isolate harvested through purification step. Therefore, RNP may or may not detectable. To generate a panel that resembles clinical sample, we conducted experiments to produce panel containing RNP by Freeze-thaw protocol. In this study, we used 3 SARS-CoV-2 isolate, which are preserved biological material belonging to the National Reference Laboratory for COVID-19. Isolation was carried out with differences in incubation time and the amount of freeze-thaw processes to see the difference of viral CD and RNP. Feral E6 cells used for virus isolation and propagation were maintained in a BSL-2 laboratory using CC plus 25 cm² and incubated for 1 to 2 days until it reached 90% constant. The SARS-CoV-2 cultures were conducted in BSL-3 facility for 3, 6, and 9-day incubation with 0, 1, 2, and 3 cycle of freeze-thaw. A total amount of 500 microliter of isolate were taken from each treatment, then extracted manually by the spin column method, while the RMP and pathogen detection conducted by real-time RT-PCR. The data obtained were then analyzed descriptively to determine the freeze-thaw process against the CD of viral and RMP gene. Based on the observation, it is known that the 3 SARS-CoV-2 virus isolates on the third-day culture incubation have shown CPE or cell morphological damage when compared to the control. The result of virus examinations using real-time RT-PCR method showed a significant increase in the number of viruses from the initial average CD 29.29 to 11.53, while the CD value did not differ significantly in additions of the 6 and 9-day incubation period. Therefore, incubation for 3 days is optimal in producing the SARS-CoV-2 virus. The freeze-thaw treatment, which is intended to break down cell tissue, is known to increase the number of viruses harvested. However, in this experiment, the CD value of the virus did not change significantly. This may be due to the majority of viral variants are already outside of the cells in the first 3 days of observation. Unlike the case with CD virus, which was not significantly different from the increase in incubation time, in this figure we can see that CD for RMP has increased with increasing incubation time. Initially, the RMP was not detected, or greater than the cutoff value. On the last day of observation, the average value was able to reach 22.66. The CD for RMP also improved in the presence of the freeze-thaw treatment. The best CD can be obtained by 2-time freeze-thaw processes. However, for the freeze-thaw treatment, it seemed that the additional freeze-thaw cycle more than 2 times did not cause a different impact up to 9 days of incubation. The limitation of the study is that we did not observe the effect of incubation time and the freeze-thaw processes earlier than 3 days and later than 9 days of incubation. Another study of the freeze-thaw aspect on RNA detection by PCR method was carried out on dengue virus by onward. The results show that the freeze-thaw process did not affect the RNA detection. As the conclusion of the study, the number of viruses produced was good enough on day 3, but the number of RMP produced up to 9 days was still in the intermediate level. Therefore, this method is promising for continuous development to obtain optimal CD. It is necessary to carry out a further study with a longer incubation time to determine the best condition for producing a balanced CD number between viral gene and RMP. The others would like to thank the directors of CRD-BBHC and all the member teams of SARS-CoV-2 virus testing at the National Reference Laboratory for COVID-19. Well, that's all for me. Hopefully this information can provide additional insight about COVID-19 and let us fight against COVID-19 together and win the final victory. Thank you.